So, now let me give you a few applications one can use by applying this technique. So, people apply 2D for various objectives. For example, studying the global protein expression. When I say global protein expression, it means you want to identify or separate all the proteins present in a given protein sample. So, if you do not have information about all of the proteins which could be present in your sample mixture, then first of all one need to create that fingerprint or blueprint of all the proteins and use that by the process known as global protein expression. Second thing, which is most commonly being used, is you want to compare the protein abundance from a given sample to a treatment sample. So, that is known as differential protein expression, or also known as abundance based proteomics. You want to look at the proteins which are either going upregulated or downregulated in their expression because of a given treatment, or because of a disease condition, or because of your experimental condition. So, in both of these cases, the two-dimensional electrophoresis can be used to resolve the proteins from very complex mixtures. People also used to study different type of isoforms or the post-translational modifications which occur in that process. One of the major advantage of this technique is the visual analysis of proteins. When you stain a gel after the two-dimensional electrophoresis process, then you can see all the proteins spot on the gel, which is not possible from the gel-free approaches such as mass spectrometry. Over there, most of your analysis depends on your spectra and you have to rely on your analysis. In this case here, all the protein spots are already present and you can visualize that, and then you can use each of these spots to further analyze and compare the images. So, the protein profiling, if you want to compare your different sample types from condition A to condition B, you need to solubilize the proteins from the cells and separate all the proteins by applying the pH which is shown here in the first dimension. After that, you need to reduce and alkylate your samples and then separate that in the SDS page based on the second dimension molecular weight. Then depending on your staining method being used, whether Coomassie brilliant blue or silver stain, different type of fluorescent stains, or autoradiography, you can visualize the protein spots. Now, one can compare these images from condition A to condition B, and if there are some proteins which are different in their expression, those can be the proteins of your interest. One need to analyze that from different type of gels, different replicates, both technical and biological, and then obtain the statistical information for these spots. And if these are significant, then this is the protein of your interest which you would like to identify by using mass spectrometry. So, I am giving you a view of abundance based proteomics or differential expression profiling, where you can separate the proteins and you can compare the abundance of each spot as shown in the 3D views, and then the spot of interest could be trypsinized and it can be identified. The peptide spectra can be generated from mass spec. Now, let us move on to the case study. In first study, we will talk about serum proteome analysis of Vivax malaria, an insight into disease pathogenesis and host response, a study by Ray et al. So, the study was performed by my group, and we have tried to investigate the host response against malarial pathogen Vivax. So, this slide gives an overview of the workflow which we followed to investigate the host response against Plasmodium vivax in human serum. So, serum samples were obtained, proteins were extracted, and the protein samples were compared from the healthy and vivax patients by applying gel based proteomics, including 2-dimensional electrophoresis and difference in gel electrophoresis. We separated large number of proteins on the gels, compared the data, looked for those proteins which are significantly differentially expressed in vivax as compared to the healthy controls, and then those proteins were subjected to mass spectrometry analysis. Further, we validated few targets and analyzed the data for various type of pathways which could be involved in such disease. So, before we move to details of various experiments performed in this study, let me show you this animation to give you an overview of the workflow followed in this experiment. So, let me give you an overview of all the steps involved in performing such an experiment. So, first of all, carefully withdraw the intravenous blood into a vacuum thinner tube and store the tube on ice to allow the blood to coagulate. Centrifuge the contents to separate coagulated blood cells and clotting factors from the serum, which forms a clear supernatant. After centrifugation is done, then collect the obtained serum in a fresh tube. Sonicate the serum to break down any large protein complexes. Serum contains several proteins in wide range of concentrations, of which albumin and immunoglobulin G are the most abundant ones. The presence of such high abundance proteins can interfere with the experiment. Therefore, we need to remove these high abundant proteins. So, in this experiment we tried both sonication and depletion of the high abundant proteins. After sonication is done, then we can add the serum sample on the columns. Usually these are commercially available, which can deplete various high abundant proteins. Now, these depletion columns bind only the high abundance proteins onto their matrix through the affinity interactions. Once the serum has been processed using a depletion column, precipitate out the remaining proteins by using trichloroacetic acid and acetone. Once the protein extraction is done, then we need to add the rehydration buffer, which contains urea, thiourea, CHAPS, DTT and BPB. After overnight incubation, then we can centrifuge and collect the supernatant. Now, we will watch the quantification of the proteins. So, prepare the Bradford dye reagent and label the tubes suitably for the standard and test samples. Add the standard and sample solutions to their respective tubes. Now, add the Bradford color reagent to the tubes and mix them thoroughly. Once the color reaction has occurred, then perform the absorbance measurement at 595 nanometer. Once you have obtained good amount of proteins which was determined by the protein quantification, now proceed for the rehydration of immobilized pH gradient strips and focusing of these rehydrated strips. So, remove the IPG strip from the cover, place it in the rehydration tray, add the reconstituted protein sample on the strip, pour mineral oil to prevent it from the drying, and then move forward for the isoelectric focusing. So, place the wicks at the either end of the IPG strip, followed by an electrode at each end. Fill all the adjacent wells with the mineral oil to ensure the uniform current flow. You can input the desired protocol on the instrument software along with the details of strip length, pH range, number of strips, etc., and start the focusing process. You can monitor the focusing progress on the software, and you need to ensure that focusing is proper. During the IF, proteins will migrate along the strip and come to rest at a point when their net charge becomes 0, which is known as their isoelectric point. After the IF is done, then we need to separate the proteins in the second dimension on SDS page. Remember, in between you have to do the equilibration strip as well. So, SDS page, which constitutes the second dimension of two-dimensional gel electrophorese, involves assembly of the gel apparatus, gel casting, equilibration of IPG strip, followed by placement of the IPG strip on the gel and protein separation. So, prepare the gel casting solution consisting of acrylamide, bisacrylamide, trischloride, SDS, APS and timid. Once the gel is polymerized, then you can add the IPG strip, and now ensure that assembly is properly sealed and buffers are properly placed, so that proteins can be separated based on their molecular weight. Depending on the gel length, you can perform the second dimension separation for few hours. After second dimension separation is done, then we can perform the staining and de-staining. So, this process involves removal of the gel from the electrophoretic assembly, followed by treatment with a fixing solution, staining solution and finally, the de-staining solution. You need to ensure that proper shaking conditions are maintained, and you need to allow 10 to 14 hours, usually overnight step for the staining as well as de-staining steps. After de-staining is done, then you can see a protein spots on the gel with the very clear background. Now, serum poses challenge of abundant proteins. So, first of all, we had to remove the abundant proteins from the serum, and there are standard columns available from various commercial manufacturing, from which one can remove selectively the very highly abundant proteins such as serum albumin, IgG and various other abundant proteins. In fact, there are columns which can deplete almost 14 abundant proteins present in the serum. Now, what should be ideal sample which one can use for such proteome experiment. So, first of all, to address this question, we tried separating the proteins from various type of samples. We took crude serum, because serum will have mostly the protein content. So, we thought, can we directly apply the serum on the IPG strip, immobilized pH gradient strip, separate the protein and see how many proteins we can separate on these 2D gels. But serum contains lot of salt. So, isoelectric focusing itself becomes challenging if you have crude serum. So, we also tried whether we remove the salt component of it, and after desalting, if we do the IEF. Since serum contains highly abundant proteins and these commercial columns which can deplete these abundant proteins selectively, those are quite costly, and considering that large number of patient sample you have to process. So, we thought, can we disrupt these abundant proteins and remove these low abundant proteins from these abundant proteins by using sonication. So, effect of sonication as well as desalting was also investigated, and then we also used commercial columns to remove the abundant proteins, followed by the desalting step. Now, sample was corrected and processed with all these 4 variables which I talked to you, and after that the standard procedure of 2-dimensional electrophoresis was followed. Now, when you are doing this serum protein separation, the IEF becomes very critical, because you have lot of salt component in it, and if your salt removal is not effective, your isoelectric focusing run may not be very smooth. So, from the softwares you can monitor how smoothly your isoelectric focusing is progressing. So, we tried various type of effects, crude serum alone or sonicated and desalted, desalted alone and depleted and desalted, and then try to compare the effect of these type of processing on overall proteome coverage. We also tried to see that how many protein we can separate on the small gels, and then as expected the small gel of 7 centimeter cannot provide us very large number of proteins, but even these gels can be used for standardization process, and as you can see, starting from the crude serum to the desalted and then finally sonicated and desalted gives us better coverage of the proteome. We tried the same with the even larger strip 11 centimeter, and it was again quite convincing. Then we moved on to the very large strips 24 centimeter for the clinical studies. So, in general we took 40 microliter of the serum, which was precipitated with the 4 volume of ice cold acetone containing 10% of TCA, incubated at minus 20 degrees, centrifuged, followed by added 1 ml of ice cold acetone to wash the precipitate. Then incubated it on the ice for 15 minutes, and again centrifugation was performed. Acetone containing supernatant was removed, and then pellet was dried in the lysis buffer. The recipe for lysis buffer we have talked earlier when we discussed about sample preparation, which is also mentioned in the slide here. You can use urea, chaps, IPG buffer, DTT and BPB. So, from the previous study we looked at replicate gels, and is 3 here, and we found that desalted, sonicated and depleted sample gave us largest number of spots on the gel as compared to crude alone or desalted alone or desalted and sonicated. So, these studies were performed on the small strips to standardize the procedure, but we are able to draw the conclusion based on the reproducible pattern obtained in these gels, and then we applied the desalted, sonicated and depleted these conditions for the processing all the samples. Now, after processing of these samples, now how to extract the proteins. So, protein extraction protocol was also optimized, and to optimize these protocols, first of all we looked at what are different methods available in the literature. So, people have applied direct crude serum, TCA acetone precipitation, sonicated and desalted serum, trizol extraction method, as well as abundant protein removal and TCA acetone precipitation. So, we used this modified procedure of TCA acetone precipitation, where we used depleted and desalted and sonicated serum. We also compared the effect of acetone alone or TCA acetone precipitation. As you can see in this slide, the left one is showing the overall protein spot present in the acetone alone treatment, and right side slide showing the TCA acetone precipitation. So, overall more number of spots were resolved on the TCA acetone precipitated gel. So, since we standardized our condition on the small gels earlier, we also try to apply that on the large gel of 24 centimeters, because that is where we have to perform all our analysis. So, we again compared crude versus depleted protein response of these serum samples on the large gel, and as expected the depleted serum sample showed more number of spots as compared to the crude serum samples. We also try to look at effects of various type of staining, whether it is Kumasi blue staining or silver staining. We definitely identified more number of spots from the silver staining, but just because of we want to compare a large number of clinical samples with the different healthy controls, it was very difficult to standardize the conditions to keep it uniform staining for the silver. So, to overcome this limitation, we use the colloidal Kumasi and biosafe Kumasi staining, and we standardize the same condition for all the gels. So, this slide gives you an overview of various type of parameters which we investigated from different type of treatments: crude serum, sonicated and desalted, TCA acetone precipitated and trizol extraction methods. So, we looked at effect of sonication, desalting, rehydration, amount of protein loading, type of strip, staining solution, how many number of spot we can resolve on these gels, and then since software analysis automated gives lot of artifacts, how many real spot we can obtain after the manual refinement. So, I am trying to give you all the details what one need to actually perform before reaching to that stage where you can compare the healthy control with the clinical samples. And this lecture is actually trying to cover various type of concepts which we have talked in the sample processing and in different lectures of two-dimensional electrophoresis. So, now after doing lot of comparative analysis and standardizing the protocols for sample preparation and protein extraction, now we are ready to perform the comparative study on serum samples of VIVAX patients with the healthy controls. This is one of the gel image showing that is what is expected on 24 centimeter large 2D gel, and then a representative gel showing the healthy control and the Plasmodium VIVAX treated samples. So, these samples, as you can see, were showing large number of protein spots resolved on these gels, and then after separating these protein samples on these 2D gels from various patients and controls, then most challenging task was the data analysis. So, automated matching as well as manual refinements are performed to obtain the how many spots are significantly modulated due to VIVAX infection. As you can see in this slide, we had looked at various controls and various VIVAX patients. These are only few patient samples here, but we analyzed a large number of samples. but those spots which were reproducible in all the patients, for example, one you can see in the top panel, 2 spots are showing significant alteration in the y-wax as compared to the healthy controls. So, these type of spots were considered further for the mass spectrometry analysis. Now, those spots which we found were showing good differential expression, we looked at the 3-dimensional views of those spots to ensure that those are not artifacts and those are the real spots showing up or down regulation of these proteins in human serum. Now, after completing the analysis, we were able to obtain large number of proteins which were differentially expressed, and as you can see in this graph, there are proteins which vary in different range of the fold change. Many proteins show very less fold change between 1 to 1.5 fold, and there are few proteins which also show more than 10 fold up or down regulation. So, these protein spots were considered interesting because those were statistically significant, and then these spots were further analyzed using mass spectrometry techniques. So, this slide gives you an overview of tandem mass spectrometry technique and followed by how LC-MS/MS data can be analyzed by using Mascot search engine. So, different type of mass spectrometry techniques are available, including MALDI-TOF and LC-MS/MS based approaches, and we will talk about these techniques in much more detail in the next module when we talk about mass spectrometry. But just to give you an overview here, that one need to analyze the spectra and also one need to have some standard proteins to ensure that correct spectra is obtained. So, you have to do a lot of quality control checks in mass spectrometry to ensure that your data is of good quality. I will cover the details of mass spectrometry experiment when we talk about mass spectrometry in more detail in the next module, and again maybe I will take this study at that time, that what are the different detailed procedure required to perform such analysis. But at the end, from these experiment of mass spectrometry, one can obtain the identity of these proteins, and as you can see in the slide, there are several proteins in the host human were differentially expressed due to the Plasmodium vivax infection, and these are some of the proteins listed here, such as haptoglobin, apolipoprotein A1, serum albumin precursor and clusterin. These were down regulated, and serum amyloid A, celloplasmin precursor, leucine rich alpha 2 glycoprotein precursor and alpha antitrypsin precursor. These proteins were upregulated among many other proteins. How two-dimensional electrophoresis technique can be applied for various biological applications. By giving you a case study and details of various type of experiments one need to perform, probably you got an idea that before performing that final experiment on your case and the controls, actually a lot of optimization has to be performed, and starting from your sample processing and how you can expand the coverage of the proteome. These type of quality control checks are very essential. If you can increase the overall proteome coverage, you can separate over 1500 to 2000 proteins on the gel reproducibly, then you have a good chance of identifying various potential targets, which could be novel drug target or potential biomarkers. But if you have not done the proper quality control experiment, you have not optimized the protocols properly and your proteome coverage is very poor on the gel, then you are comparing only partial proteome, and there is a good likelihood that you will miss out many important changes. So, not only today we discussed about one application, how host serum protein changes due to one of the Plasmodium parasite, but also we discussed the various nitty-gritty experiments one need to perform to achieve such type of comparison. We will continue our discussion on some more applications of two-dimensional electrophoresis and also two-dimensional difference in gel electrophoresis, because due to the variations in the 2DE, people also try two-dimensional Dij experiments, and also that is very sensitive. So, that also expands the overall proteome coverage. So, we will continue our discussion in the next class. Thank you.